stimulation of the alcohol dehydrogenase gene in Drosophila melanogaster, fruit fly, using optogenetics, by Tressa Coultard. Background. Optogenetics is a new field that combines optics and genetics to control defined events in specific cells of living tissue. It was developed by Carl de Souza and his colleagues at Stanford University in 2006, and then was named Method of the Year in 2010 by the research journal Nature Methods. This new field of study is very exciting because researchers are now allowed to control specific processes in specific cells in specific areas of the body in freely moving animals. Part of optogenetics is the opsin, and opsin is a light responsive channel that allows certain light into the cell. So there are two types of opsins. You have microbial opsins and animal opsins, and in this project we are using the first type, microbial opsins. One of the microbial opsins is a channel rhodopsin, and we are using channel rhodopsin 2, which responds to blue light. And then we have the GAL4 UAS system. The GAL4 UAS system is used to turn the ADH gene on when it is activated by the CHR2 channel rhodopsin. The UAS is an upstream activation sequence which specifically binds to the GAL4, and the GAL4 is specifically bound to the ADH gene. And so when the UAS upstream activation sequence goes up and binds to GAL4, the GAL4 activates the ADH gene. The ADH gene is alcohol dehydrogenase. The alcohol dehydrogenase gene breaks down the harmful alcohol that is in our body into smaller, lesser molecules that is converted into energy and used by our surrounding cells. So the hypothesis of this research is if there is no alcohol to be digested, is there any RNA produced, even if the gene is activated by light? The hypothesis was that there should be no RNA produced, for there is no alcohol in the fly system. Experimental groups. So we have 3 groups in this. We have the GAL4 UAS Drosophila, which is our manipulated group, and this group has the GAL4 and the UAS, which means that their genes should be turned on by the blue light. Then we have 2 control groups. We have the GAL4 Drosophila and the UAS Drosophila. GAL4 and UAS by itself does not affect any of the processes in the cell. So all of the flies were kept in the same cabinet, were fed at the same time, were fed with formula 424 instant Drosophila medium, and when anesthetic was needed, were given fly nap. Our procedures. So to begin, we had to cross the Drosophila because they only came as GAL4 and UAS. So we had to cross them to get our GAL4 UAS and to get our manipulated variable. So 1 GAL4 infected male was placed with 1 UAS infected female in their own vial, and this process was repeated so we had about 10 vials. Then, after they had created offspring, we then took the offspring and put them into their own vials, so we had a clearly marked GAL4 UAS group. After this was done, we then exposed all 3 groups to the blue LED light. This is to activate the ADH gene in the GAL4 UAS system group, and while we did it to that group, we had to do it to the other groups just to keep it with continuity. We then waited 3 hours after we exposed them to the blue light. After the 3 hours, we then extracted the RNA and we converted it to cDNA. So, we extracted the RNA by using RNaQES for PCR-Q, which includes a series of washes to clean off all of the tissue to just expose the RNA at the bottom. And then the RNA was then converted to cDNA using a TACMAN reverse transcription reagents kit, which involves running a PCR reaction, which amplifies it for us. After we had converted it to cDNA, we then wanted to measure the gene expression. So we used a real-time PCR machine, which includes a custom assay for the ADH gene. And after it was ran for about 3 hours, we then had our results. So this is the amplification plot. And as you can see, we have our 18S and our ADH. We have an 18S because this is a control variable. All cells basically have an 18S gene, and so it's a good control to have. Then we have and that is green, and then our ADH gene is purple, the gene we are seeing test. So as you can see, that they all went and they all amplified. So our RNA extraction was a success, and our amplification was a success for gene expression. Also, the 18S hits the line, the CT line, first because there's always more 18S inside the cell. This is our relative quantitative, seeing how much gene expression comparing to all samples. As you can see, there wasn't much gene expression in any of the samples, and the gene was not expressed very much. Analysis. The RNA extraction was a success. This was deduced from good nanodrop readings that we had taken after the conversion to cDNA. And then after the real-time PCR reaction was ran and the cDNA was amplified, with the graphs we have seen, we could see that the amplification was a success due to graph 1 and graph 2. However, in the relative quantification graph, there is not much gene expression, so the ADH gene was not expressed, as shown in the second graph. Conclusion. The hypothesis stated that no gene expression should occur since there is no alcohol in the fly system, and this research proved that right. However, if I wanted to change this, I should have had a positive and a negative control, showing the normal amount of gene expression in flies when they have alcohol in their system. Instead of withholding alcohol from the flies, I should have given them a set amount to see what levels of gene expression this gene produces when activated naturally. I should also have given, I maybe should have given my experimental group to see if maybe more RNA was produced when they were artificially and naturally stimulated. Acknowledgements. I would like to thank the following: my teacher, Henry Weeks, for having such a great program going on at my school and to always being there when I need help. Stephen Pulver, he introduced me to the Gal4UAS system and helped me develop my hypothesis. Backyard Brains for developing the optogenetics machine, the blue LED light that I used to deliver the light to the groups, and Lindsay Giffey, my fellow student who helped me learn about RNA extraction and gene expression.